Hi everyone, welcome to Reason with Science. I'm your host Jitendra. This episode is with Pavel Plevka. He is a research group leader at Central European Institute of Technology. His research is focused on solving the structures of viruses in order to understand their mechanism of action. For this conversation, I'm also joined by Anna Papageorgiou, a friend and a colleague. Here we talk about general aspects of viruses, their genomes, evolution of viruses, their infection strategies, impact of viruses on human health, environment, and agriculture. Enjoy the conversation, share and subscribe to support the podcast. Thank you for listening. Hi, Pavel. Welcome to the podcast.
Hi, Pavel. Welcome.
Hello.
Yeah, so as a scientist who's been working in the field for many years now, let's start with something basic, a basic question like what is a virus and why do we study them?
So virus is a form of life that has two forms of existence. It exists as a virus particle, which has like a compact shape. It has a protective surface layer, and inside it carries a genome, the genetic information of the virus. And then when it enters a cell, it causes infection, and it uses resources of this cell to produce the virus progeny.
Okay, so basically, also you say that like the building blocks of viruses are like the nucleic acid that it brings, the genetic material, which is being coated by a capsid. And in some cases also there are like extra somehow layers, extra membranes, but we will talk about all this later, right?
Yes, absolutely.
Okay.
Yeah, so interesting thing here is that, so when we think of viruses, there is a huge diversity there. Starting with, we can simply start with what is the extreme of viruses like in terms of size. So, so what are your comments here?
So viruses as a group are extremely variable, as you said. So the smallest viruses will be as small as 30 nanometers in diameter, while the largest can exceed more than 1 micrometer. And this also defines like the huge range of genetic material that they can carry. So the smallest viruses are extremely simple. They will encode just a few proteins, like the capsid protein that forms the shell, and then a protein that enables replication of the virus genome. While the most complex ones, they carry hundreds of genes that perform various functions, and they can be to some extent independent of the cellular metabolism of the cellular functions. And your initial question was about the variation in the type of genetic material. So unlike the cellular organisms who store their crucial genetic information in the form of DNA, viruses can also use RNA, and then they can use also various types of the RNA. So it can be double-stranded or single-stranded, and the same goes for the DNA. Then when they carry just the single strand, it can be also positive and negative sense, meaning that it may, if it is the positive sense, it can directly serve as mRNA for protein synthesis, or it can be the negative strand that needs to be copied into the complementary version before it can be used for protein synthesis.
Okay, but then regarding the two extremes of the like viral size spectrum, for example, from one part we may have giant viruses, and then from the other part we may have subviral agents like viroids or gene transfer agents. So could you a little bit introduce us to this kind of extremes?
Mm-hmm. So there are also like forms, let's say, of life that are even simpler than viruses, which would be, for example, the viroids that you mentioned. So these are just small segments of RNA that are able to trick the RNA polymerase of a cell to make copies of themselves. So these can often be found in plants where they can cause actually very severe infections. And then if you look at this like infectious agents, so sometimes prions, like the proteins that can have several conformations, one that is functional for the cell, and another one that is more stable and toxic, and then can trigger other proteins to adopt its conformation are also assigned to the infectious agents somehow related to viruses.
Okay, so despite that size somehow reigns, like even giant viruses, I think that they are at the absolute, limit of what can be visualized by a conventional light microscope, right? So given that limitation of size, how can we visualize viruses? What are basically the tools that you as a structural biologist are using to study them?
Mm-hmm. So indeed, the largest viruses are about micrometer in size. So they can be just barely detected using the light microscopy, but definitely we cannot distinguish any details of their structure. So traditionally, viruses are studied using electron microscopy, so where you can comfortably see the virus particles. And to find details of their structure, for a long time, the method of choice was X-ray crystallography, because the viruses, the particles have very regular structures. So they can actually form crystals and then can be analyzed using this technique. Nowadays, with the development of cryo-electron microscopy, it's definitely the method of choice to look at virus particles. And it enables studies of even more complex viruses like bacteriophages that combine the icosahedral symmetry of the head and then they have a helical tail with some appendages. So this all can be studied very nicely using cryo-electron microscopy. And electron microscopy also enables us to look at the infected cells, because the virus infection will induce reorganization of the infected cell. And we can observe also the assembly of the new viruses inside cells.
So as you already mentioned about the size and shape of the viral particles, and there is a lot of variability there as well, right? Like all the viruses, they have a different shape, or it can be different size as well. So what are the different shapes there? If you can just introduce some of them and some of the viral systems that you are studying, how different they are.
Yes. So besides the differences in the size, there are differences in their structure, and there is this typical classification of viruses. So the two basic types are viruses that have icosahedral capsids and helical capsids. So these viruses, they use symmetry to be able to utilize one type of protein to assemble complete shell that will protect the genome, which is much bigger than the information it encodes. So using icosahedral symmetry or helical symmetry with just one type of protein, the genome can be protected. And then there are some more complex viruses, like the mentioned bacteriophages, which can combine the icosahedral symmetry for the head and then helical symmetry for the tail. And also there are the asymmetric viruses, which are often membrane enveloped, like the infamous influenza or HIV or coronaviruses.
Basically, that's there is incredible variety, from bacteriophages that they look somehow, if I can comment on that, like interplanetary probes, or or some virus like influenza that they're more or less spherical, or polyhedral viruses like adenoviruses, or even filamentous ones like Ebola. Really, they are quite diverse in terms of shapes and structures, but with such morphological somehow diversity, because you have already introduced us that they do bring different genetic material, they do bring different structure shapes, they have different size. How do we classify viruses, basically? Because I think to study them you need really a classification system, right? It helps, or maybe more, I don't know.
So traditionally, there is this well-established Baltimore classification of viruses that is based on the genome they contain. So it's classified according to the type, whether it's RNA or DNA, and whether it's single-stranded or double-stranded, and positive or negative sense. And it also includes these reverse transcribing viruses, which are known as retroviruses. That actually during their life cycle convert the genome from DNA to RNA form and then back again.
So one of the exciting things about all the viruses is that the fact that they can infect their host, right? So let's start talking about that. That how do viruses recognize their host, and how do they infect them?
So yeah, as the viruses differ a lot in their structures, and of course the hosts are also very different. So the hosts can be bacteria, algae, but also higher eukaryotes like humans. So the viruses need very different approaches to be able to infect these cells. But there are several like common topics. So usually the viruses at their surface are equipped with proteins that recognize something from the surface of the host cell. So that's called a receptor. So initially the virus will bind to a receptor, and then this triggers some changes in the virus particle or in the host cell. So in many cases, the host cell itself engulfs the virus. There is this process called endocytosis, when the virus is delivered to the inside of the cell, but it's still enveloped by a membrane that is derived from the cytoplasmatic membrane of the cell. And then the viruses need to somehow cross this membrane. And there are two major groups of viruses that use different approaches. So when these are enveloped viruses, they will fuse the envelope from the virus particle with the endosome membrane, and the content of the virus spills into the cytoplasm and then can trigger infection. When we have non-enveloped viruses, they have to somehow induce disintegration of this endosome membrane so that the virus particle can enter cytoplasm. And yet another example, for example, can be considered as the bacteriophages that actually attach to the surface of bacteria, and then they somehow puncture a hole through the bacterial cell wall and cell membrane and eject their genomes, just the double-stranded DNA genomes, into the bacteria to induce the infection.
Yeah, just so that people, they get a taste of it. I mean, I remember that you had those 3D models. If we maybe can just like show it, and people can get an idea that, okay, how do those particles look?
Yes, indeed. So I have two examples here. So the black one would be example of a bacteriophage. So this is a virus that would bind to the bacterial surface like this, and then the tail would actually extend a little bit through conformational changes and through addition of some proteins that are at the beginning inside of the phage head. So it extends, then it crosses the bacterial cell wall, and then the DNA that is stored inside the phage head, they're under pressure, it's ejected into the bacteria. So this is nice example of the delivery. And this is another virus. So this is actually a model of a virus that would infect honeybees. So these are like the higher eukaryotes. And we can observe in electron microscopes that particles of this virus, they attach to the surface of a cell, and then they are engulfed into the endosome, as I mentioned before. So they end up in membrane-bound vesicle. And then the cell actually tears apart the endosome membrane, and the virus particle is released into the cytoplasm, where the capsid cracks. It opens like a Pac-Man into two halves, and the genome from the inside diffuses out into the cytoplasm. And these are positive sense RNA viruses. So the RNA itself can recruit the host ribosome that will start production of viral proteins.
Okay, just to continue, like on what we were discussing slightly before. So you said, okay, the virus, it managed to infect the cell, so it's now in the cytoplasm. And you said that it triggers infection, right? So what does it mean? What is coming next?
So when the genetic information of the virus reaches the cytoplasm, and for some of the viruses, the genetic information needs to reach the cell nucleus. This is typically for viruses that have DNA genomes. So then the genetic information is, if it is DNA viruses, it's transcribed into RNA form and then translated into proteins. And mostly the proteins execute the functions of the virus, and the early functions are to take control of the host cell. So in many cases, the viruses will shut down the host proteosynthesis, or they will at least limit the innate immune responses of the cell, so that they can start taking over control of the cell and sort of push forward production of additional viral proteins that will enable replication of the viral genomes. And once sufficient number of the genomes are copied, the, let's say, later phase of the infection progresses, when the proteins that form the virus particles are formed, and the assembly is initiated. So then once the, like, for example, for the bacteriophages, the capsid will form without the tail. The phage DNA is packaged into the capsid under high pressure, and then the tail is attached. For these RNA viruses, the viral RNA will condense, and then the capsid assembles around it. And after this, the viruses have different strategies. So, for example, the bacteriophages, they will induce lysis of the infected cell. So the cell basically bursts, and many of the viral progeny particles are released to the environment. These small RNA viruses, they either induce lysis of the host cell, or they are exocytosed in membrane vesicles. While other viruses, typically the membrane-bound vesicles, viruses, they can bud from the membrane. And this way, the infected cell can survive for a long time, still continuously producing the virus particles.
Yeah, this is really fascinating that these tiny particles, like after all the struggle, they enter in a host cell, and then, they have to like kind of get everything synthesized and get assembled and then go out of the cell. So how much of this mechanism we know?
Well, so as we started our discussion, so viruses are very diverse, and some of them are better studied than others. And because they are actually so different, even in the like molecular mechanisms of their replication, so many things cannot be generalized from virus to virus. But of course, many viruses, in particular the like important human pathogens, are studied in great detail. So we know many like molecular details, how the viral proteins not only form the particles, but how they also interact with the host cell, how they take over control of the host cell. For other viruses that are less studied, we know close to nothing.
Yeah. So did someone manage to kind of study this assembly of the viral particles in vitro setup, like try to do it in Eppendorf? Are there any studies?
So for some viruses, the assembly can be reproduced in vitro, and you can form the particles in an experimental tube. Whereas for many other viruses, this cannot be reproduced. So the assembly is essentially dependent on some cellular structures that cannot easily be reproduced in vitro. So typically for bacteriophages, the head assembly is initiated from the inner bacterial membrane. This is difficult to reproduce.
And just one small question here. So what about like viruses, like for example, influenza, that they do bring segmented nucleic acid, like they do bring, for example, 8 pieces of nucleic acids that they need in the end to be assembled, in the intact new viral particle. How did they manage to, to do this?
So yes, so this is very interesting question because as you mentioned, influenza has segmented genome, and to have productive infection, the cell needs to receive like all the segments. So we have this combinatorics problem that the particle needs to pick up all the different segments to be infectious. So the viruses either can have a specific mechanism where they recruit all the genome segments into one particle, or you can have this variant that there are actually multiple copies of each segment. So this will increase the probability that each is represented at least once. Or the viruses can also rely on the fact that to infect a cell, not only one virus particle will enter, but multiple will enter.
So now, like at short time spans, viruses are usually limited to infecting one host species. But how do viruses cross this like species barrier and are able to jump from one host to another? How do they manage that?
Mm-hmm. So, yeah, as you mentioned, usually viruses are specific, and this jumping of the host barrier is rare. But it can be easier for some viruses. For example, for influenza, that can be made easier by sort of combination of genome segments from different viruses. So in rare cases, in rare unfortunate cases, there can be like co-infection of one cell by two different viruses. And then these segments are reassorted, which could sort of expand the host range of the virus. And these are the cases, for example, when the influenza strain is called like a bird flu. So this would presumably have some proteins from influenza that normally infect birds, or the swine flu from viruses that infect pigs. For other viruses that have non-segmented genomes, this like crossing of the species barrier usually requires mutation. So if, like, let's say, non-human virus is to infect a human, usually the infection will be very inefficient. But then in the human body, some mutations of the virus can occur, and they can be more efficient in infecting the human cells. So basically, in the infected individual, there will be this selection of the virus to adapt to be more efficient in infecting human cell. And if the virus acquires favorable combination of mutations, it can adapt and establish itself as new pathogen.
Okay. And since you were talking about genomes, I mean, it's also interesting that nowadays in biology we talk a lot about information about genomes, what is there. So how much of this information about like how viral particles they do what they do, like how much of it is encoded in the genome or in the genes? What do viral genes they encode?
So what are the essential parts? So the virus is defined by the virus particle, and the shell, like the surface of the virus, of the simplest viruses, is formed by proteins. So this type of protein is encoded by the virus, and then inside is the genetic information that needs to be copied in the host cell. So the simplest viruses can encode just the capsid protein that will form the particle, and then the replicase that will ensure copying of the viral genome in the infected cell. So that will be just two genes. But many viruses carry additional genes. So for example, the capsid can be more complex, like for the bacteriophage, it can be made from many different proteins. And also inside the cell, the viruses can take advantage of more functions than just copying its genome. So to take efficiently control over the host cell, they can have multiple proteins that will, degrade parts of the cell or alter the cellular functions.
Yeah, and also like there are like a lot of viruses that they have an additional like layer covering the capsid, and this is usually like lipid layer, right? So can you tell us about like the nature, the composition of this layer, and also like the origin of those basically membranes? From where are they coming?
Mm-hmm. So indeed, so some virus particles can be covered like in a bag by membrane layer, and this membrane is derived from a cellular membrane, but it can be from different parts of the cell. So some viruses, they will bud from the cell membrane, so from the cell surface, but other viruses can bud into, for example, into endoplasmatic reticulum. So that's another source of the membrane. And besides acquiring the membrane, the viruses also insert their own proteins into the membrane that covers the virus particle. So it's the famous spike protein of coronaviruses, for example, that is embedded in the membrane and then recognizes the host cell. And usually the viruses will include like several types of proteins into their membrane. And these proteins can be responsible for different functions. So it can be attachment to the host cell, like recognition of the surface features of the host cell, but also this membrane fusion. So when the viruses are enveloped in the membrane, they will deliver the content of the virus particle into the cell by membrane fusion. So the membrane from the virus will fuse with the membrane from a cell, and the content spills into the cytoplasm.
Mm-hmm. So the existence of membranes, it just helps in the in kind of the infection strategy, like in the endocytosis, right?
Yes. So it helps with the infection process, but also in a way it makes the virus vulnerable, because the membrane will be sensitive to detergents, for example. So that's why the membrane envelope viruses are relatively easier to destroy. While those that have only the protein capsid, for example, they can dry out and then still remain quite infectious.
Yeah, this is really fascinating. And so, I mean, another thing that I remember is, as Dobzhansky said, nothing makes sense in biology except in the light of evolution. So let's a little bit talk about evolutionary origins of virus. Mm-hmm. So were there only single origin, or there were multiple origins?
Mm-hmm. Yes, indeed. So the virus origins and virus evolution, that's very interesting, a little bit philosophical question. Nevertheless, as the viruses are extremely diverse, it is likely that they are polyphyletic, meaning that they have multiple origins throughout the evolution. And then generally, studying history of viruses is difficult because they are too small and they don't leave any fossils behind. So we can just like have some theories and infer things from trying to find out relationships amongst viruses. So one of the like possible, or there are several theories how viruses may have emerged. And one of them is that actually the sort of parasitic form of life emerged very early in the evolution of the life itself. Actually, maybe even before the cells, before the life acquired the form of cells. So at the very beginning, there was probably this RNA world of self-replicating molecules. But already at this stage, it is possible to envision that there were some molecules that didn't have the ability to copy themselves, but could be copied by other molecules. And this would be exactly this parasitic form of life that is now common to viruses. Another alternative how viruses may have emerged is by simplification of cellular parasites. So we can imagine that there is some like cellular form that becomes intracellular parasite of another organism, and then it's convenient for this parasite to become very simple, so that it doesn't impose big burden on the host cell and can sort of multiply efficiently. And as this parasitic organism is getting simpler and simpler, it may lose many of its functions and depend just on the host. And eventually we could end up with something as a virus. And third theory is that viruses emerged as sort of like a function of a cell that became a hostile. So there are these gene transfer agents that are particles that enable exchange of genetic material between some bacteria. And these are actually very similar to bacteriophages, and may, if these particles acquire the ability to preferentially transfer the DNA that encodes their functions, they would actually become a bacteriophage.
And now, like a little bit, to elaborate a little bit more, why basically are viruses not placed on the tree of life, even though they do infect like bacteria, archaea, and eukaryotes, all life forms?
Mm-hmm. Well, so they are exactly not placed in the tree of life because they didn't originate, like they do not share like the same origin with the living creatures. So they do not fit into the tree. So they are like detached branches, let's say. So they do not fit into the scheme of the cellular organisms.
And how does it make you feel? No, just kidding.
How does it make you feel?
So, I mean, talking in the same down the same lines. I mean, so the point is that the evolution of species, it doesn't happen like alone. There is always co-evolution, right? And the same thing should go for both viruses and their hosts. So let's talk a little bit about co-evolution of viruses and hosts.
Mm-hmm. Yes. So this is very nice question. Thank you. So of course there is long co-evolution. So there is arms race between viruses and their hosts. So the hosts, they were forced to develop immune system to defend against viruses. And over the time, viruses are developing mechanisms how to neutralize the immune system so that they can infect the host. And there is very advanced warfare. So like the human immune system is extremely complex. It has many components like the non-specific and specific immunity. And this adaptive immunity can respond on various levels. At the same time, viruses carry functions to neutralize the immune response. And as we learned with coronavirus, for example, even though normally our immune response can last for a long time, so with the SARS-CoV-2, we are experiences these reinfections after a relatively short time. So they somehow managed to avoid the long-lasting immune response. Another example is the co-evolution of bacteriophages and bacteria, where the bacteria developed so far more than 70 identified types of immune system that block the phage infection. But phages are responding in kind, and in many of them, more than half of the genes inside the phage genome are probably devoted into fighting these immune systems of bacteria.
Probably that's the reason why they exist, right? Because if they won't be able to kind of breach the bacterial immune system, they won't exist anymore.
Yes, absolutely. So on the other hand, there is also this aspect of viruses contributing to the evolution of the cellular organisms. So in some rare cases, some genes from viruses or bacteriophages become integrated into the host genome, and they can provide additional functions.
Yeah. So basically also like comment here, this is a way that geneticists nowadays, like they trace also the evolutionary history of viruses, especially for those they can be integrated in like genomes. And it was interesting, like in this ENCODE project that it was released in 2010, if I remember correctly, where we got to know, for example, that 8% of like our genome, of human genome, it's coming from, it has viral origins. Mm-hmm. But if we focus a little bit on prokaryotes, so we know that prokaryotes like bacteria and archaea, they have an immune system that protects them against viruses, and this is called CRISPR. Mm-hmm. But also we know it has been also found that basically I think nowadays that viruses possess also anti-CRISPR systems. So could you elaborate a bit on this kind of like systems and how did they co-evolve? Briefly, maybe.
Mm-hmm. Yes. So the CRISPR-Cas systems. So that's a type of adaptive immunity for bacteria. So when infected, the bacteria can take a segment of the infecting bacteriophage and incorporate it into their genome. And then they have a system that will target this sequence. So whenever the bacteria is infected by this phage, it will cleave its genome before the phage can establish the infection. And that's how the bacteria is protected. And this is carried over to the bacterial progeny. So the future generations are also protected. So that's very nice for bacteria. But of course, bacteriophages develop various approaches to avoid this mechanism. So they can have special proteins that will interfere with function of this CRISPR system, or they try to mutate away, so they do not contain the sequences that are targeted by this system. And yeah, indeed, this is a complex arms race. So another example that we benefited from, because the CRISPR-Cas system is now becoming a tool in molecular biology, but even better established tools are restriction enzymes. So these are actually also results of the arms race between bacteriophages and bacteria, where the bacteria developed enzymes that will target specific sequences which are not in the bacterial genome. And then the phages are developing various mechanisms to avoid this degradation, for example, by modifying their nucleic acids.
Yeah, I think that's a good example of or the evolutionary arms race. But then once we are talking about co-evolution, we can also talk about gene transfer agents and like in prokaryotes specifically. And one of the study that you guys did, which was this gene transfer agents and of Rhodobacter. So if you can please discuss.
So yes, thank you. So we looked at the gene transfer agent, which is basically a particle that in structure looks very similar to a small bacteriophage. And it's called extracellular organelle. So how do the Rhodobacter bacteria use it? So when there is this like population of cells and they start to be in unfavorable conditions, for example, they start to starve, they have a system that detects the cell density. And then in the population, about 1% of cells, they decide to commit a suicide in a controlled way. So they will start producing these gene transfer agents, and then the bacteria will segment their own genome and package small segments of it into this gene transfer agent. And then this DNA is delivered into the bacteria that are around. And probably this helps the bacteria to survive because it enables recombination of DNA from different bacteria. And by this recombination, the bacteria may gain new function that will enable it to survive in the environment, like to utilize new source of carbon or some other element or some source of energy. So these systems are actually very common among bacteria living in oceans.
Okay, so far you have, I think, given us a lot of information about viruses. Could you please tell us how widespread viruses really are, just to have an idea?
Yes, so viruses are indeed very widespread. So first we can look at bacteriophages. So many of us acknowledge the fact that like bacteria are everywhere. They are on all sorts of surfaces. And when we want to prepare food, we need to wash it and clean it, not to get infected. However, of course, these bacteria can be and are infected by bacteriophages. So it means that the phages are basically everywhere where the bacteria can be, and it's everywhere. But the good thing is that, as we discussed at the very beginning, the phages are quite specific. So they do not only infect many bacterial species, but they pose no danger to us. So if we eat them, it's just extra protein and extra DNA. And when looking at sort of higher eukaryotes like us, so each species can be infected by many types, many species of viruses. So, and those are produced in millions and billions in the infected organism. There are many virus particles around.
Cool. So it seems like we live in a cloud of viruses, if we could say.
So
okay.
Yeah. Virologists are happy to say that we live in virosphere.
Yeah.
So whenever we say that I have a gut feeling or something, it may be just bacteriophages infecting gut bacteria or something. So,
yes. So, yeah, there are some studies, of course, that show that the gut bacteria are very important to determine many aspects of our life, like how healthy we are, even maybe impact our moods. But those populations of bacteria in our gut are controlled by phage infections. So this way, even the phages can have some impact on our life.
Okay, so now that we have the basic information about viruses, let's focus on viruses infecting humans. And we know that many human diseases are caused by viruses, from like common flu to fatal diseases, right? So one problem here is that we cannot target viruses with antibiotics, like how we are dealing with bacteria. And also we do have a lot of antibiotics, but not drugs for to deal with viruses. So why is it like that? And basically, which are the therapeutic strategies that we can use to deal basically with viruses?
So generally, antivirals are a bit more difficult to develop than antibiotics because viruses are simple, and for their replication they use cellular functions, which of course cannot be targeted because we would kill the host cell. And there's another aspect that bacteria differ in many aspects of their life from humans. So there are a number of bacterial functions that can be targeted that will not affect humans. So for antibiotics, we have quite a few targets in bacteria, but for viruses, we have only few functions that are executed by the virus that can be targeted. And typical target that is valid for all viruses is the enzyme that enables replication of the virus genome. So this can be targeted by analogs of nucleotides. So often we take advantage of the fact that the viral replication proteins do not have the proofreading activity. So we can provide analog of a nucleotide that is then incorporated into the virus genome. But because of the proofreading abilities of the human replicases, this is not incorporated into the human genome. So then this will induce excessive mutation of the viral genome, and the virus population will collapse. And there can be numerous other targets. So the viruses, for example, can carry specific proteases, proteins that will cleave other viral proteins to enable maturation of the viral particles. So one very famous target is the protease of HIV that is targeted by numerous successful antivirals.
Yeah, this is really interesting. So of course, we can do all that. And the other thing is that simply prevention is better than cure. So what are those prevention strategies that we can use?
So yes, prevention is usually cheaper and better than curing the disease. So very strong tool that we have is vaccination, of course. And vaccination has been developed against many viruses, and we take advantage of the fact that actually our immune system is predisposed to react very strongly to virus particles. So when we have a viral particle, it's made from the capsid protein that is actually repeated at least 60 times over the viral surface. And our immune system is designed to react very aggressively against these repetitive surfaces. So one approach to produce the vaccines is to have particles of a virus that do not contain the genome, for example, that are then injected into the vaccinated individual, and the immune system will learn how to react to these particles and then protect against infection by the real virus. There are other approaches to develop vaccines as well.
And now, if we may ask, like, how does your work contribute in finding therapeutics that target viruses?
Okay, so my group is focused on basic research. So we are characterizing the structure of viruses. So we can use cryo-electron microscopy to determine the structure of the virus with atomic resolution, which means basically we know where individual amino acids are. And this structure can serve for design of some compounds that will bind to the viral particle and, for example, prevent it from opening. So with these small compounds attached, the viruses will not be able to induce infection. And other contribution is that we look at how these particles infect the host cells. So we describe the mechanism that the viruses use to enter the cell and to deliver the genome into the cytoplasm. And again, this may open possibilities to block this process and thus prevent the virus infection.
since we were discussing about antibiotic resistance, etc., and viral particles being a great delivery tool, and from your work, I mean, the structures of viruses, they are also a good portal to understand alternative ways of targeting some diseases, right? So one of the therapy which, again, I remember, and you also work on it, is phage delivery. So how does this phage therapy, how does this method works? And what are the different phages that you are using, or if there are different approaches?
So yeah, indeed, when phages were discovered, which is about 100 years ago, the scientists immediately realized that they may be used to cure bacterial diseases. There were initial attempts to use them for this purpose. However, later antibiotics were discovered, and they became the primary therapeutic option. Now, with the onset of common antimicrobial resistance, the phages are coming into interest again. And the idea is that when a phage replicates in a bacteria, it kills it. And that's the way to eliminate bacteria even from human organism. So in this direction, we are studying bacteriophages that infect Staphylococcus aureus and Pseudomonas aeruginosa, which are two human pathogens. And our aim is to describe how phages infect individual bacterial cells and how they efficiently produce the phage progeny. But also in the human body, when the bacteria cause infection, they often form biofilm, which is like a surface layer covered by bacteria that are embedded in so-called extracellular matrix that they produce. And this matrix protects the bacteria from the immune system. But bacteriophages, because they co-evolved with the bacteria, as we discussed, so bacteriophages, they developed mechanisms how to infect bacteria even inside biofilm. So the aim is to sort of identify bacteriophages and describe the mechanisms that enable them to efficiently infect bacteria in biofilm.
And yeah, it's great that at least now we can use like phage therapy as a solution to tackle the problem of antibiotic resistance, as you nicely mentioned. So now another promising use of viruses is in cancer therapy. So can you introduce us to the novel therapies that are using basically viruses as anti-cancer agents?
Well, so I would describe like two things. So viruses can help like with development of vaccines. So because they provide this repetitive surface, so we can attach like specific, let's say, proteins or epitopes to the surface of the virus particle. And then when these particles are used for vaccination, they will trigger strong immune response against the epitopes, the proteins that we added to the surface of the virus particle. So in this way, viruses can help us with development of vaccines. But also the virus particles can be targeted against the cancer cells. So this can take even advantage of the fact that the viruses have properties to efficiently replicate in the cancer cells and thus kill them. So it's a combination of these properties to be like targeted to the cancer cells and then to eliminate them.
Yeah. So I mean, another important use of viruses can be especially in genetic diseases. I mean, we were talking about CRISPR, and CRISPR is increasing in a way to edit the genome of the newborns, or even like at the embryo stage, to kind of remove the genes which are problematic or which may cause a genetic disease. So how we can use viral particles in this gene therapy?
Yes. So during evolution, the virus particles developed to be able to enter into cell and to deliver the genetic information that is inside. So this can be taken advantage of, as you mentioned, by replacing the viral genome inside with some specific information that we want to deliver into the cell. So in this way, the viral capsids become a tool in the development of therapeutics. So it's not only necessarily about removing some cellular function. The viral particles can also deliver like the desired information, genetic information that is missing in cells of the affected individual.
So just a comment here, basically. I think we tend to view viruses only as disease-causing agents in general. And indeed, they are responsible for a lot of disease, like in humans, plants, and animals. But also, from our discussion, we understand that there exist potential benefits that viruses can provide to their hosts. And initially, we talk about like this coevolution of virus and their hosts, and so the diversity, and how basically viruses, they promote like diversity, one stuff. And now also we discuss about basically how viruses contribute to human health, like how we can use them in different ways to tackle diseases and etc.
Yeah, the other use can be in the ecological and environment sense, that since we were talking about coevolution, and viruses do infect bacteria, archaea, fungi. So how basically viruses they are important for ecology, and how do they affect the biogeochemical cycles?
So yes, so viruses can have the form, for example, of like biopesticides. So they can be used to control populations of some pests in a quite a natural way. But what you are asking about, if whether the viruses can have significant impact on our planet. So indeed, they can. So for example, the algae that grow in the ocean, so that they can, in some cases, grow into very high population densities. By that, they will even change color of the seawater, and this affects how much heat from the sun is absorbed by the ocean. And so when the algae grow, after a few weeks, it's called the bloom. This algal bloom is terminated by some virus infection. Because if the hosts are plentiful, eventually there will be some virus that will be able to infect all these cells, and the whole bloom that can cover like hundreds of square kilometers, even thousands of square kilometers, is terminated by the virus infection. And then, of course, like the bodies or remnants of the dead algae sink to the bottom, which will remove nutrients and also like carbon dioxide in the form of calcite from the atmosphere and from the sea environment. And this in turn further affects the climate.
It affects what then?
It affects climate.
Climate,
Yeah.
yes. Yeah. So now basically you describe like the example of Emiliania Huxley, this algae, and how it's being infected by a giant virus. I think it is called Phycotina virus, something, if I'm correct. Okay. And if I understood correctly, so it's like carbon fixation by Emiliania blooms is an atmospheric carbon dioxide sink, right? Or no?
Yeah, indeed, over the history. This to explain it a little bit more. So Emiliania Huxley is an algae that produces coccoliths, which are calcite discs that covers surface of this algae. And over like millions of years, these calcite discs they detach from the cells, or when the cell die, they will sink to the ocean bottom, and then they accumulate in huge quantities and basically transform into limestone. And the calcite disc, they for the algae to produce the calcite, it uses calcium and then carbon dioxide, which is fixed in this form and indeed removed from the atmosphere.
Yeah, this is interesting. And when we are thinking about existence of viruses, I mean, and they do infect other hosts. So how do they exist in the extreme conditions? I mean, the fact that some archaea, they can exist like even at minus or even at 80 degrees or even at lower temperatures. So how do they survive these extreme conditions?
Yes. So from the studies, it turns out that whenever they are living cells, the viruses will follow. So even cells that live in extreme conditions, like 80 degrees and very acid environment, like pH 2 or even lower, they can be infected by viruses or bacteriophages. And then the phages, they adapt to these conditions by having very stable particles. So the capsids, they can indeed withstand these extreme conditions. And they can do this by strengthening the interactions between the capsid proteins. So in some of these bacteriophages, for example, they will have the proteins, the capsid proteins connected in rings, like the chainmail armor of the ancient knights.
That's fascinating. And so, I mean, so thinking about, the viruses existing in this extreme environment, and like even in your work, you've shown that, for example, change in pH may kind of collapse the viral particles, etc. So then how, for example, they have to be, so the capsid should be too strong to even like avoid these kind of, even like to avoid some small changes in the environment. And also, what about like the pressure exerted by the genome, which also you kind of talk about in your work?
So the virus capsid has to complete like two basic functions. It has to be able to protect the virus genome in the environment, which can be more difficult in this extreme environments. But also, it needs to be able to ensure that the genome is delivered into the host cell to initiate the infection. And the viruses just have to have both these functions, otherwise they would not survive. And then this delivery of the genome into the host cell can be mediated by various mechanisms. That's what we discussed before. So, for example, for the bacteriophages in these extreme environments, the capsid itself can be very stable because the genome is delivered through the tail. So the head doesn't need to disassemble; it can be very stable. But for viruses that infect our cells, so usually the viral particle will be engulfed by the cell and then delivered into the endosome, and then it needs to reach the cytoplasm. In that case, the virus particle needs to be able to sense that it's inside the cell. The capsid then needs to open to release the genome. And the viruses can have various mechanisms for that. So, for example, they can sense changes in pH because the endosomes are acidified, but also changes in ion composition because the composition of the solution outside of the cell and inside of the cell is different. There are different ions. So this change in the ion composition can trigger like decrease of the interaction of the strength of the interactions between the capsid proteins.
Yeah, and another work that you are doing is on honeybee viruses. So why do you study them?
Why do we study them? So when I was deciding what projects to tackle in my group, I was kind of looking at important viruses, and it turns out that like the population of honeybees is suffering from numerous virus infections. So like almost all honeybees that exist around are infected not with just one virus, but multiple viruses. And it's the virus infections that are causing collapses of the bee colonies, in particular in winter. Yes, and then I realized that none of these viruses were characterized before, so the structures were completely unknown. And also in Brno, where I work, so there is this tradition of Mendel, and at Mendel University in Brno, they have apidology department where they study honeybees. So I found a very good collaborator, Docent Antonín Přidal, who could help us with production of the honeybee viruses, which is difficult because there are no tissue cultures that could be used for the honeybee virus production. So we have to use pupae. We have to infect individual pupae and then use them for isolation of these viruses. And this enabled the subsequent structural studies.
Okay. So we also have, I think in one question you have already answered, but we have like two questions, I think, like out of curiosity. It's like if we can characterize them a little bit speculative questions. So do viruses encode genes with a known function? Especially these like giant viruses that you're studying and where I think we don't know much about them, right? What do they bring in their genome?
Yes, indeed. So the more complex virus, the longer the genome, the more functions it carry. And as we discussed at the very beginning, so some viruses we understand quite well. These are mostly the human pathogens and simple viruses, but some bacteriophages or some of these giant viruses that you mentioned, they can have the genomes as long as several mega base pairs, and they can encode hundreds of proteins. So many of these will be uncharacterized and with completely unknown function. It doesn't mean that they are not important for the virus. It's just that for now, we do not know why they are important.
Okay.
Yeah, so the next question is, I think it's quite philosophical. Most asked question probably about viruses. So are viruses alive?
But you already started, from this question, which is our last one.
Yes, so this is more like philosophical question because viruses certainly do not care whether they are alive or not, but for the human definition. So in my view, viruses are alive at the stage when they infect the host cell. Because when a virus infects the host cell, the cell usually doesn't have very bright future ahead of it. It will serve just production of the virus progeny. And at this stage, the virus will take all the cell resources for producing the new particles. And I think it can be considered to be alive. When it exists as the individual virion, the virus particle, then it doesn't have many attributes of life.
Okay. Now, it's just like one more question. So what still our technology lacks? I mean, what we still can't answer in the field of virology?
So with the current development in cryo-EM and with the many previous studies by X-ray crystallography, the structure of individual virus particles has been characterized in great detail. So we understand fairly well this extracellular stage of the virus. But then when a virus infects a cell, the situation becomes extremely complex. So even if it is a simple virus, it interacts with thousands of various proteins and functions inside of the infected cell, and it needs to take control of the host cell. And this is without distinction. We never understand this completely for any virus, even the most studied ones. So there is still plenty to learn.
Okay, good.
Yeah, and what is your favorite virus?
What's my favorite virus? I have to say that my favorite viruses are bacteriophages because they are like beautiful molecular machines. So in the evolution, they gained the ability to infect bacteria using their tail. So in particular, the tailed bacteriophages, which actually can contract, and they have various devices to attach to the bacteria and to drill through their surface layers.
And okay, now that we close the session, if we may ask you, do you have any suggested literature for us and also the audience, other than, of course, your lab's publication?
So there is one article that I'd like to recommend that is by Kasper and Klug, and it, basically it's not an experimental paper, but it provides reasoning why viruses will have icosahedral or helical symmetry. So it's in the early days when the electron microscopy wasn't like strong enough to really resolve the details of the virus particles. But just by thinking about the viral genomes and how the particles look like, they determined that they will have icosahedral symmetry, because it enables efficient use of the coding capacity of the viral genome to create a protein protective shell.
I think it's quite fascinating, and all this, like, to talk about viruses, to get to know about like how do they work and etc. It's amazing, and I mean, alive or not alive, they certainly impact our lives and the life of the planet.
Exactly.
So I think with that, thank you so much for accepting the invitation and shedding the light on our crucial part of our life on viruses through your work. So thank you so much.
Thanks a lot, Pavel.
Thank you very much. This was a pleasure.